Hello from Chem Help ASAP. In this video, we will walk through a general overview of what happens and when in terms of preclinical safety during a drug discovery and development program. Upon completion of this video, students will be able to list many of the common types of studies that fall under the umbrella of preclinical safety and distinguish between efficacy and safety studies. Preclinical safety studies are performed throughout both drug discovery and drug development stages. Because preclinical testing occurs both in drug discovery, which is truly preclinical, and drug development, which is after the drug advances into the clinic, sometimes preclinical safety is called non-clinical safety. Preclinical safety studies typically begin with in vitro assays. Target selectivity is a common factor when the intended target is a member of family proteins. The desired final drug should inhibit one member of the family, preferentially over other family members. This type of selectivity can be tested through binding studies. Sometimes computational in silico methods are also helpful. Secondary pharmacology refers to off-target compound activity on targets that are frequently associated with adverse effects. Phototoxicity, which is an increase in light sensitivity, and genotoxicity, which involves DNA damage, can both be monitored through preliminary in vitro assays. Short-term toxicology and safety pharmacology require in vivo studies in preclinical species. The data from all these studies help determine the safe starting dose for human trials. Once a lead advances into the clinic, phase 1 trials begin with short-term safety studies. In parallel, chronic toxicology studies in animals will be initiated to support the safety of the compound for longer dosing regimens that may be required in phase 2 trials. Similarly, reproductive and developmental toxicology studies will also be started. Finally, if a drug will be dosed chronically, carcinogenicity studies will be necessary. All drug assays and studies may seem the same. All the studies are monitoring for a change, whether the change is in vitro or in vivo. The two types of activity, efficacy and safety, however, are viewed very differently. Efficacy studies for a drug program are designed to measure the benefit of the drug. Later efficacy studies often hinge on animal disease models. These are diseased animals that are treated by a compound to see if the compound can improve the animal's condition. The disease models will be specific based on the goals of the drug program. In contrast, all drugs must satisfy safety criteria. So, compounds of interest will be tested for a range of safety risks, generally using the exact same assays and studies across different drug programs. In some cases, the mechanism of action of a drug may call for additional testing. For example, a compound that acts on the liver may have additional studies focused on liver toxicity. Safety studies involve healthy animals. The goal is to monitor whether a treatment leads to a safety risk for a healthy animal. We have listed some representative types of studies that are part of a preclinical safety program. We have also differentiated between efficacy studies and safety studies. That was a very quick overview of a preclinical safety program. The following videos in this playlist will go into more detail on specific aspects of preclinical safety. Take care.